Thanks for joining us on this very rainy Friday. This is actually super excited. And today we're going to have a lecture by Professor Adrian Figg, who's coming to visit us from Virginia Tech. So they're basically trading good weather for horrible.
It was raining when I left too, so I brought the rain then. I think that's how it goes. Yeah, sorry, my bad.
So Adrian's been an assistant prof since 2021. Prior to that, we met in the Merkin group. So he did his postdoctoral study with Chad Merkin at Northwestern. He was basically the polymer boss of the entire group. And so that was pretty much every polymer question's like, yes, please help us, Adrian. This is the way. And so I'm really excited to see what came up of this. And also from PhD, he does his PhD at the University of Florida with Professor Brett Summerlin, where I think that was probably the genesis of a lot of the controlled radical polymerization.
Undergrad, actually.
Yeah, you have like a lot of undergrad, like it's insane. No, but he worked with Bert Meyer, Patricia Dankers, and Craig Hawker, as well as Brett Forrest. Sorry, pronunciation. But yes, like that seems to me like a whole overlay of everything polymer coming together that led to the PhD, that led to postdoc, and now the career. I think this is going to be great. I also saw an excerpt, I think, of this talk maybe a year ago as the work was being done, and he totally owned it. That was super exciting. So looking forward to see what happens since then. And with this, I will stop talking and leave the floor to you.
Thank you. Yeah, thanks for the introduction. It's always fun to visit your friends that you've known and sort of gone through the whole academic process with. Catherine and I used to sit right next to each other for about 2 years. So I didn't know anything about oligonucleotides, so she was the one that was telling me oligonucleotides and helping me troubleshoot the synthesizer. So it's cool seeing all that. I haven't really spent a lot of time in Canada, so it's been really cool coming across the border and seeing what's going on at McMaster. I've had a great day so far in the meetings. 30-minute meetings could sometimes be like a blessing or a curse, and every single one of them has gone over because I've just been enjoying the conversation so much. So it's been a really enjoyable day. And I'm excited to tell you what my group has been doing for the last 4.5 years at Virginia Tech, where mostly what we do is we look at new ways, we develop synthetic methodologies to think about acrylic copolymers and maybe either develop ways to synthesize them differently or just think about the structures differently. So if you need to know anything about me, I obsessively think about controlled polymerizations. Every aspect of my academic career has been thinking about controlled polymerizations. And I just want to ground us on what I mean when I say controlled polymerizations, just so as I'm going through some of the data and some of the slides, you know what I mean when I'm talking about that. So if we take the set of monomers in maroon and the initiators in orange, these are the Virginia Tech colors if you're thinking that's a weird color combination. So if the monomers are maroon and the initiators are orange, in a controlled polymerization, we essentially want to start all of our chains at the same time. And if we start them at the same time, that means they're all going to grow approximately the same rate. And so what this means is if they grow at approximately the same rate and they start at the same time, you'll have this low molar mass dispersity because they're all growing at about the same rate. So you'll overall have the same average molecular weight. What's also really important is that you get these living chain ends. So after you do this polymerization, you have minimal termination events so that you could include, say, this blue monomer and make this block copolymer. This is really the power of controlled polymerization, is being able to make block copolymers or more complex architectures because they'll have inherently different properties than the two homopolymers if they aren't covalently attached to each other. So that hits us this living chain ends and these defined block copolymers. And then also what's really important is this predictable degrees of polymerization. As chemists, this is what we want, right? We want to be able to know, if you add this amount of stoichiometry and you run this reaction for this long, this is going to be your molecular weight or your output. This is really important as a chemist to be able to predict what your product is going to be. And so how we do this is essentially you get a linear increase in molecular weight with conversion if your polymerization is controlled, which is great. So these are just sort of the qualities that we're looking for in the polymerizations. The reason why I love controlled polymerizations is this is my favorite example of why it's so interesting and important. So if we take ethylene, which is a very simple molecule, right, C2H4, and you do an uncontrolled polymerization, so a free radical polymerization of ethylene, you're going to get, this is a topology of the polymer structure, so this would be polyethylene. You're usually going to get this linear low density polyethylene with all of these branching points. And then this is what we use for garbage bags, 6-pack rings, that's low density polyethylene. We all sort of know what that material property is like. Alternatively, if you do a controlled polymerization, in this case using a metallocene catalyst, you're going to get this linear polyethylene structure. So this is a repeat unit. So you're having over 100,000 ethylene units covalently attached to each other in a single chain with no branch points. And so ultra-high molecular weight polyethylene is what the US military uses for like bulletproof siding in vehicles. If you say get a hip replacement, they're gonna put polyethylene in you. It's a very tough material. Even though both of these are just polyethylene, they have the exact same sort of chemical makeup of carbon and hydrogen. We're just changing the way that we're connecting the monomers together, and that's drastically changing the properties. So my group doesn't work with polyethylene, but we love this inspiration of like, if we can think about connectivity and composition, maybe we can take sort of the same monomers and just the way that we connect them, or if we can develop a new way to connect them, we're gonna get drastically different properties than conventional ways that you could connect these types of monomers. And so we do that with acrylic polymerizations instead of polyethylene. That's where I'm really interested in for the controlled polymerizations. So the technique we do to do this is called reversible addition fragmentation chain transfer polymerization. I like kind of getting a read on the room on who's familiar with it. So who is familiar with raft polymerization in general? Oh, great. This is actually more than usual. So of the people that raised your hand, if I asked you to come to the whiteboard and draw the mechanism, how comfortable? One, maybe? Two? Okay, three, great. All right, that's actually way higher than usual. Usually people are like, I don't want to do that. I'm not going to ask you, don't worry. So a lot of people are familiar with the technique or have heard of it. I like going through the chemistry because my group thinks really mechanistically. Like we look at the mechanism and think of what are the challenges in the mechanism, how do we develop new synthetic methods that will help us overcome these challenges, how do we use these polymers. So I'm just going to walk through the mechanism and then tell you sort of where we innovate through the mechanism. Raft starts with this conventional initiation pathway, usually. So this could be like a thermal initiator like AIBN, where you heat it up and then it thermally degrades. You lose nitrogen and you can get two initiating fragments. These initiating radicals are going to react with monomer in solution to give you this prepolymer. And then this prepolymer is going to react with this. This is what we call the raft agent. It's a thiocarbonyl thiol compound. So these are specifically designed to mediate the polymerization. So what we want is we want this prepolymer that was initiated by the initiator to react across this carbon-sulfur bond to give us this intermediate. So we're going to call this a chain transfer event. And then these raft agents are specifically designed to fragment off the R group in this sort of pre-initiation stage. So we design it so the equilibrium goes this way, and this R dot is more stable than this PN dot. And so just to like ground you in some stoichiometry, usually with initiators, we want about 1% relative to chain transfer agent. So that means 99% of our chains are going to be started from this R dot, and only a small percentage are going to be started from this initiation pathway. We're just using that to funnel radicals into the solution. But a majority of our chains are going to be initiated from this R dot, where we specifically designed this raft agent to fragment this way. After this sort of small molecule raft agent is consumed through this R dot, or this R dot can go on to polymerize and create more monomer, and then it's going to go activate more small molecule polymerizations. After the small molecule raft agent is consumed, this PM could just be any sort of growing polymer chain. It can go in solution. We're going to have this thiocarbonyl thiol compound on the end of every propagating chain. So this active radical can undergo this chain transfer event, and then it can kick off this PN, and now we get this PN propagating radical. But the important thing is that in the main propagation step, those radicals are thermodynamically equivalent. It's essentially the same monomers, they're thermoneutral. So we'd call this a degenerative chain transfer process where like if you're polymerizing polystyrene, this would be a polystyrene radical and this would be a polystyrene radical. And really the raft agent is just helping those growing polymer chains share the low concentration of radicals, so they're all growing at about the same rate. And then because this is radical chemistry, they're all going to terminate through conventional radical termination events. So that's like the chemical description of raft. I like using metaphors a lot. So I'm going to use a metaphor to like help you understand what the raft agent is actually doing. Because I think it's really helpful because the mechanism can be confusing, and even like as a grad student, it took me a while to like fully internalize it and digest it. So the metaphor is if we were in this room for an hour and like having a discussion, and my job as a mediator was to like walk a microphone around so that everyone that wanted to talk had an opportunity to talk. If I just like gave the microphone to Catherine and let her talk for an hour, my job as a mediator, I'm not doing a very good job in my mediator position because you got to talk for an hour and no one else got to talk. Instead, if I'm like keeping track of the time and like making sure that everyone has an equal opportunity to grow as the mediator, I'm just running the microphone around, but everyone has about the same amount of time to talk. So it was equally shared. And that's sort of what the raft agent is doing. There's like a low concentration of radicals, and it's just sharing those radicals around through all these chains. They all grow for about the same amount of time over the reaction course, the course of the reaction. So does that help understand it better. It just is shuffling radicals like a hot potato, but its job is to just move them around. And so my group likes to think about all the different steps of raft polymerization. My talk is kind of split into the initiation pathway, the propagation pathway, and then what we do with the polymer. I've actually decreased this section quite a bit lately because I'm really excited about the polymer section. So I'm just going to tell you the different mode of initiation that we use predominantly in my group. So we actually use what's called photoelectron energy transfer, or PET initiation. So in exogenous initiation, this could be some sort of azo initiator, where you're relying on like heat to degrade that azo initiator to introduce radicals into the solution. You're always going to have some amount of chain ends that were started from that thermal initiator, so you get like a lower purity of polymer chains, let's say. What we do is we get some sort of photocatalyst that we can excite with light. The excited photocatalyst can undergo either an electron or an energy transfer reaction with the thiocarbonyl thiol. In this case, I'm showing an energy transfer because we end with two radicals. So after this energy transfer, we initiate, and this can propagate, and then you'll get a deactivation step where these radicals just recombine to give you your starting raft agent. So we don't have to have an initiator that goes to that initial initial chain transfer process to initiate our chains. We're just relying on the photocatalyst to react with the chain ends and have some sort of activation, deactivation. Those chains can also do the hot potato mechanism. They're not kept away from doing that. We're just introducing the radicals in a different pathway. And so like the conventional advantages are that you start all of your chains from your raft agent, you can control the photocatalyst concentration and radical concentration. These are what we're really interested in my group, is thinking about how, conventionally you run a polymerization according to how you're introducing radicals and the Kp of the monomer. But what's really nice about this photocatalytic technique is that you can actually decouple radical introduction from temperature. So my group likes to take advantage of reaction kinetics to sort of access different sorts of structures. So if you can access a different energy barrier by changing the temperature, but you're not changing the concentration of radicals in the solution, you can think about synthesizing more precise structures by like playing with the kinetics of what's going on in solution, while not changing anything else except for just the temperature and how that temperature is affecting the reaction pathway. You're not changing radical concentration or anything else. So we love that. That's what we think about a lot. The other idea is that you can really readily control radical concentration via the catalyst and the light intensity. So again, like if you have a pretty high energy intermediate that can degrade with a pretty low concentration of radicals around, you can tune radical concentration pretty readily, just changing photocatalyst, and it's a pretty direct input compared to some initiating systems. And then the other idea that we're starting to get really excited about is that there's a ton of different catalysts that span absorption wavelengths and reduction in potential energy transfer, and the energy of the reduction or the energy transfer. So then you can start thinking about like these complex structures where you can think about absorption wavelength correlating to like a reduction potential and start to get this sort of architecturing of a macromolecule according to like the wavelength. I'm not really going to talk about a lot of these. These are just like where we're going in the future, and I'm going to show you sort of the introduction as we're ramping up into these areas of macromolecular synthesis. So for the rest of the talk, I'm going to focus a lot on this propagation step and why some challenges in polymer synthesis result from this degenerative chain transfer step. And then the last half, I'm really excited to talk about like what you can actually do with some of these polymers. So where the story begins today is from this JAX paper that we published a couple of years ago. In this paper, we were actually trying to put exactly one vinyl ether, or like one monomer at a defined position within an acrylic polymer. This is challenging because usually like radical polymerization follows a Poisson growth distribution, so it's like a statistical likelihood of adding monomers. You can't be like, I want one monomer at this place and only one monomer at this place and then like chain extend it. That's really challenging to do in polymerization. We were able to do that again by playing with kinetics and temperature to make sure that we were only getting one addition of this monomer at a defined position. We're really excited about it. But what's really interesting is like I show this step where we're going from a benzyl vinyl ether to a methyl acrylate polymerization. This looks really easy to draw on paper. It's sort of just like a raft polymerization of methyl acrylate when you first look at it. But like I could draw anything in ChemDraw and say like this is going to work really well, right? This is actually really, really challenging and goes against like the way that raft polymerization is designed. So we're going from this benzyl vinyl ether, which is a pretty high energy radical, and then we're going to try and chain extend it with this methyl acrylate, which is a relatively more stable radical than the benzyl vinyl ether. We're able to do this because these thiocarbonyl thiols can absorb UV light. So if we just blast it with UV light, they'll undergo photolysis. homolytic cleavage, and then you can just chain extend them with methyl acrylate. You see, this is our single unit insertion. We can control the molecular weight after that just based off concentration, and we can get a low dispersity of our second block because we're starting with a low dispersity polymer. But one thing that's really notable is that you lose this raft agent. So if you wanted to insert another single unit addition, you don't actually have that livingness that we need for a controlled polymerization after this second chain extension. So we can put one monomer to define position, but we can't put two because we're just getting a bunch of termination during this chain extension. This is based off some of Brent Summerlin's work. We were able to do it because of this work that was developed, where they were taking xanthates now. So in the last slide, I was showing you a trithiocarbonate where this is a sulfur. You can take a xanthate. They have different absorption properties, different radical energies, but these xanthates more readily undergo homolytic cleavage. You can use UV light to, again, do this homolytic cleavage. And now the less activated monomer, or the less stable radical, is this methyl acrylate. And we're trying to chain extend this with this methyl methacrylate, which forms a more stable radical, which goes against conventional raft blocking efficiency, which I'll get into when I talk about the thermodynamics. But they were able to just homolytically cleave this through UV light, and then they can make this block copolymer of methyl acrylate and methyl methacrylate. They actually lose a lot of the chain end also, even though I have it drawn here, but you can chain extend it. The big issues with this is that you lose the chain end retention, and then this only works for xanthates. If you ever do raft polymerization, or you want to get interested in doing raft polymerization, xanthates are very rare in like conventional raft polymerizations that you do because they don't lead to as good of control due to some of the energetics. Oftentimes you'll be doing that trithiocarbonate that I showed on the last one. Like if you go to Sigma Aldrich and are like, I want to do raft polymerization, it's going to tell you to do a trithiocarbonate. This technique doesn't work with trithiocarbonates at all. So like the most common raft agent by far, it's really challenging to do. We're able to do it because we were just like blasting it with UV light to get it to go, but it's really hard to control. All right, so I want to go back to this sort of idea of the initiation process and the propagation process just to ground us in like chemically why this is so hard to do. So this is the conventional initiation process, which I'm going to use to guide us through sort of a conventional chain extension and why it's hard. So if you were doing a chain extension of methyl acrylate with methyl methacrylate, and we look at this sort of conventional initiator, if this is AIBN, we're going to get an AIBN fragment, which I have shown here. What we want to do is we want to chain extend this polymethyl acrylate with methyl methacrylate. So if you're just doing this in a conventional raft polymerization, you'd get this AIBN fragment. This is going to react with methyl methacrylate monomer in solution just based off concentration of things in solution. So any conventional chain extension in raft, you're going to get a small oligomer of the second block, like a small homopolymer of the second block, regardless of what you do. So you're going to get an oligomer of methyl methacrylate, and then this oligomer of methyl methacrylate radical is going to have to add into this polymethyl acrylate that you're actually trying to chain extend. You want to go through this chain transfer process and kick off this methyl acrylate so that you can actually chain extend all your polymers. Does anyone have any ideas why this wouldn't work? I was like putting people on the spot for this. I usually put faculty on the spot for this because it's more fun for me. Catherine? Yeah. So if we look at this, what we want to do is get this secondary radical, but we're trying to go through this chain transfer process, which is an equilibrium process, to go from a tertiary radical to a secondary radical. So the tertiary radical is thermodynamically more stable, and we're trying to get to this thermodynamically less stable product. So it actually doesn't work because of the thermodynamics. So we did DFT calculations just to show this more tangibly. So this is still that middle chain transfer process. So this is where we're going to start in the energy diagram. So we have this radical. What we want to do is fragment off to get this PMA radical. We can see that the thermodynamics are relatively thermoneutral, and then we have this energy barrier of 16.8 kcals per mole. But if we compare that to the other pathway of just fragmenting off this methyl methacrylate fragment, we see a lower energy barrier, and it's also more thermodynamically favorable because of this tertiary radical. So essentially, like, you can't chain extend a less activated monomer with a more activated monomer in raft polymerization because the kinetics and the thermodynamics are fighting against you. So it just like doesn't work, right? That just is not a thing that works. You really have to think about a different way to overcome this process, which we're relying on to actually give us control of the polymerization. So it's really challenging, and it's a problem that's persisted in raft polymerization since its inception. I've been talking about raft polymerization. I love raft polymerization, but I think it's important to like take a step back and think about other types of polymerization techniques, and if you could access this structure in any other way, because like at the end of the day, if you just want a polymer, use the technique that's going to give you that polymer. You don't have to be stuck on one sort of polymerization technique. So if I asked you to make a controlled polymerization of some block copolymer and have it really well defined, you're probably going to think about anionic polymerization first. If you do like gel permeation chromatography, all of your standards are made anionically. It gives you really low dispersity molecular weights. You can control them very well. You actually can't synthesize these types of acrylic, methacrylic block copolymers using anionic polymerization. So this was investigated over the years, but this is one example from 1998 where they're trying to make this block copolymer, and then we see that it's always bimodal when we make this. So that means that we're not getting efficient chain extensions because it's bimodal. And that's because you're propagating anion with the methyl acrylate on the chain end isn't very sterically bulky, and it's actually getting alkylated by the monomer. So you're actually losing your anionic chain end because you just have monomer around and it's reacting on the ester of the monomer instead of propagating through. So you actually lose your chain end retention and you get this multimodal block copolymer trace. So anionically, you actually can't synthesize these sorts of acrylic, methacrylic block copolymers. You could also think about ATRP, which is just another flavor of controlled radical polymerization. Chris Matiaszewski showed this in 1998, going from this polymethyl acrylate to methyl methacrylate. You can do this, so what you do is you synthesize your acrylate with this bromine chain end, and then you do a halogen exchange to a chlorine, and then based off like bond association energies and some kinetics, you can do this flip to methyl methacrylate, but you can't go back. So you can go one way, but you cannot flip the other way. You have to do some chain end functionalizations, which usually lose the chain end anyways. And so no one's actually shown that you can go make like a tri-block of like a acrylic, methacrylic, acrylic using HRP, because once you flip it, it's just harder to work with. If we look more recently, you can make these sorts of acrylic, methacrylic block copolymers using Lewis acid catalysis. So this was developed by Eugene Chen at Colorado State, where you can just mix these monomers together, and then according to the Lewis acid that you're using and their reactivity ratios, you can get these pretty well-defined multi-block copolymers. And the nice thing is that you don't have to, you can just mix all the monomers together and then just according to the reactivity ratios, you'll get this block copolymer. The challenge with this though is that as soon as you open your flask, you're gonna lose your chain end. So like if I ask my students to make a library of 50 polymers where you're just changing like the second block, they would have to make all 50 of those block copolymers individually, and then you're gonna have some dispersity in your first block because they're not all made from the same starting polymer. So it just creates some issues if you're trying to like diversify your library. So that's great, but my students wouldn't be very happy. Ideally, like you start with one parent polymer and then you can derivatize that parent polymer to see like the effects of the second block. If we look at raft polymerization, this is the example that I just showed. The only other example that is really present is from Mode and Zetterlund, where they can make these multi-block copolymers, and they get pretty good shifting in molecular weight to the right. And you can see they go from a methacrylate to a styrenic, to an acrylate, to a styrenic, to a methacrylate. They actually trick the system though. They don't make discrete block copolymers. They add in a little bit of comonomer on each chain extension to like help overcome that equilibrium. So like you add in a little bit of the next monomer to make sure that there's enough chain ends that can propagate so the energies actually match during the reaction. And if you're trying to do a technique like lithography that relies on like really precise interfaces, you're not going to get precise interfaces doing that copolymerization technique because you're actually getting these sort of gradient copolymers. And then I showed you with raft that you actually can't use xanthates and you lose the chain end. So we just wanted to think about all of this and come up with a different way to try and address this challenge that's been around since the invention of the technique, because the technique relies on this sort of equilibrium. So that's why we use PET catalysis. So the idea is that you can have your photocatalyst, and then you can choose your photocatalyst to excite light, and then according to either the energy transfer or the reduction potential, you can pair it with the chain end where you can get this activation, deactivation. This initiation pathway is distinct from that chain transfer pathway, right? Because we're just directly activating our chains with the catalyst. We're not going through that chain transfer. And so what we did is we did some calculations. So this is the amount of energy that iridium pipi has once it gets excited. It's known that iridium pipi undergoes energy transfer with these sorts of raft agents. So we're looking at the amount of energy for energy transfer. So that's 56.2 kcals per mole. If we look at our methyl acrylate chain end bond association energy versus our methyl methacrylate chain end bond association energy, obviously this bond association energy is lower because we're forming a tertiary radical. So it's a more stable radical, the bond association energy is lower. But what's important is that both of these are quite a bit lower than the amount of energy that the iridium actually has for energy transfer. So our hypothesis was that if we use iridium PIPI as a photocatalyst, because its amount of energy available for energy transfer is so much higher than either one of these, it's essentially going to be agnostic to whatever the chain end is. It's just going to get excited and then start and react with whatever polymer it finds. It's not going to be as selective because we're so much over either one of those energy, bond association energies. So this slide is just showing some controls. So here's our PMA chain end, and we want to chain extend it with methyl methacrylate. And so this black size exclusion chromatogram is our starting polymer. This is showing elution time on SEC, so like big things elute sooner, so we want to shift to the left. So if we just do a conventional thermal polymerization, which is the purple trace, you see it's very ugly and you don't want a bimodal distribution unless you want a bimodal distribution in your GPC, right? So this is not what we want. We don't want a bimodal distribution in our GPC, but this is just a conventional AIBN initiation. If we do that photo interferter technique by the Summerlin group using UV irradiation that doesn't just completely blast everything and kill everything, you still get this bimodal distribution because it just doesn't work as well with the trithiocarbonate. However, this is one of the early PET runs that we did. With PET, we actually get some decrease in this molecular weight distribution. It's broad. There might be a little bit of this peak under this sort of broad peak. But the big thing is that we're seeing a shift of the distribution. Sure, it's broad. I just told you that it's kinetically and thermodynamically difficult to do this, so we're expecting it to be broad. But it is one of the first times that we can actually show that we can like predictably control this shift and overcome that thermodynamic and kinetic limitation by going through a different mechanism. So we're essentially just going through the PET to activate, and we can actually extend our methyl acrylate with methyl methacrylate. This is really exciting because in the single unit monomer insertion paper that I talked about, it was really, we could get predictable molecular weights, but it was just based off monomer concentration. Now we can look at conversion versus molecular weight. So predictable molecular weight is one of the things that we want for controlled polymerizations. And what we see is we actually get pretty linear increases in molecular weights. So now we can better predict the molecular weight of these chain extensions. This is a pseudo first order kinetics plot. We want it to be linear, and if we see deviations from linearity, that means that we're, in this case, we're losing radical concentrations. So we're getting termination events after about 3 hours. But it's linear for the first 3 hours, so that's actually pretty interesting to us. And if we look at the size exclusion chromatograms, we see them shifting to lower elution times, and this peak is going down over time. So all of this data is showing us that under the right conditions, which I'm glossing over, but I'll talk about in a minute, we can actually get like relatively controlled polymerizations. Are they perfect and beautiful? No. But again, this is the first time that you've been able to do this with raft polymerization that has been around since 1998. Well, earlier than that, but it was named raft polymerization in 1998. So we're really excited about this. Some things to point out, these conditions probably look really weird to you, even if you don't do raft polymerization. They are really weird because we essentially had to add in a reducing agent. We're using higher iridium concentrations than usual. This concentration of polymer is about 10 times less than you'd usually run a polymerization, a raft polymerization, and we're doing these at 40. So that's where we're really trying to think about how to think about reaction conditions to change the kinetics of the pathways that are going on. So we want to limit termination. So how do we limit termination while increasing the activation of these chain ends? So that's how we came up with these conditions, is really thinking about like, what are the kinetics that we want to happen and how do we avoid some of the other reactions that we don't want to happen? I'm not going to get into all the details of that, but we're really excited about it. And we actually just had a paper accepted at Macromolecules yesterday that goes more into like the detail. We did this very extensive analysis on like the conditions and understanding this more. So if you love fundamental chemistry, that's great. We're basically over with that part of the talk. If you don't love fundamental chemistry, we're gonna like pull out of the fundamental chemistry and think about like, why would anyone care about being able to do this, right? Because I showed you all of these slides and it's like really hard to do. People have been trying to do it, like what's the point? Like, does it actually matter? So what we did is we made these block copolymers. So this is a bifunctional raft agent where we have our methyl methacrylate on the inside and then our methyl acrylate on the outside. So this is going in a conventional blocking order. And then we used our chemistry to synthesize a block copolymer that has the same composition, so the same weight fraction of methyl methacrylate and methyl acrylate. The molecular weights are about the same, so they look chemically identical. The only difference is that we're flipping the order of the blocks using our chemistry. So on this side, the methyl acrylates are on the inside, the methyl methacrylates are on the outside. Everything else looks the same. If you took an NMR, they'd look identical. We're just changing orientation. And so we did tensile testing. And so the conventional block copolymer is very brittle. So you can see it doesn't strain a lot. It breaks around 30 megapascals. It's quite brittle. If we just flip the order, now we're getting something that looks a lot more elastomeric, which is really interesting because compositionally they're identical. We're just changing connectivity to get different properties. This is really cool. I'm being honest, the big thing is that we're just synthesizing a thermoplastic elastomer in the second example, and in the first one it's not a thermoplastic elastomer. So these properties are expected, but this type of structure is very hard to synthesize using a lot of controlled polymerization techniques. The advantage that raft polymerization has over any other polymerization technique, except for maybe anionic, is that you can synthesize really high molecular weights. So in this case, we did our chemistry to synthesize a polymer that was 564,000 grams per mole, again with our reversed order. And so now this is like 5 times bigger than the last polymer that I showed you, and then we did tensile testing to see the effects. And so what's really cool here is that so this reverse one, we're going to about 90% strain, and then it breaks. With the high molecular weight, you can see we're getting up to like 1,800% strain. So this thing is stretching 18 times its length. We're getting some strain hardening, which is really interesting and we don't understand. We're trying to look at it now, but we don't understand it. And the overall stress is lower, but we're stretching these out like huge amounts. Usually you don't see acrylic thermoplastic elastomers that can stretch like this while still being like relatively tough. They're not super tough, but they're relatively tough. And what's also really cool is that they're transparent. They're a little yellow from the iridium here, but we actually couldn't break it. So this is actually it slipping out of the clamps. So before stretching, it's transparent. We can stretch it. This is to about 10 times its length. You just release the bottom clamp and it will go back to its original shape. So it has like some shape memory type thing that we're looking into, or shape recovery, not shape memory, shape recovery. So it'll return back to about its same length, and after 10 minutes, it's maybe like a millimeter longer and it's still transparent. So this is really interesting to us because like these properties are super weird, and no one's been able to study these sorts of thermoplastic, like acrylic thermoplastic elastomers, especially because they're linear architectures. Jimmy Mays was doing some, these are called like super elastomers. Jimmy Mays in like the 2000s and 2010s were working on some super elastomer acrylic polymers, but they were using these really unique monomers and these branched structures. We're just using what I like to call like dumb monomers. So like methyl acrylate and methyl methacrylate are the most straightforward monomers that you can buy from Sigma and like use them for polymerizations. And we're getting these properties. So we're really interested to think about like what other sorts of acrylic and methacrylic monomers we can use with these block copolymer structures to get other sorts of physical properties. So that's what we're looking at now. The big thing that I didn't talk about, I motivated this entire thing saying we want chain end retention. We actually can't retain the trithiocarbonate at the end of these polymerizations. That macromolecules paper that I mentioned is getting towards that, like we're getting better and better, but it's still really hard to do. So we're not quite there yet, but we're figuring out how to get there and we're working on it. But even just flipping it and having really good control on how to flip it is leading to properties that are very different from any other sort of thermoplastic elastomer. In particular, acrylic thermoplastic elastomer that's been synthesized before. So we're really excited about that. All right, I'm gonna transition to the second half of the talk now, where now it's like, okay, we can use raft to synthesize these polymers. We get this polymer out, what do we actually do with it? Like, what are we gonna use this polymer for? And so my group, one side thinks about synthetic methodology and high performance materials, which we talked about. The other side of my group likes to think about proteins and thinking about how to interface polymers with proteins. So if we think about how we currently design molecules and materials that target proteins, if you know the binding site, it's great, right? You can design small molecules that can bind to specific proteins and inhibit an interaction. In this case, this MDM2 is a protein that's implicated in cancer. Using this Nutland 3A molecule, you can inhibit a protein-protein interaction, which can help with cancer therapeutics. This is essentially like classic medchem, where you know the binding site and you're thinking about synthetic organic chemistry techniques, or now using like AI and machine learning to develop strong binders for that specific binding site. More recently, PROTACs have been very popular, where again, if you know the binding site, you can conjugate 2 molecules that bind to that binding site, or one that could induce ubiquitination. So maybe there's a protein you want to get rid of, and then you can have this binding site to a protein that will ubiquitinate this protein and degrade it. So you can decrease concentration of protein in the cell if that protein is leading to disease. Again, you need to know the binding sites for these to be able to synthesize the PROTACs chimeras. Also really recent, or not recently, but peptides are also very popular. Ozempic, I think now is the most talked about one, at least in America, where you can have this peptide sequence. So this is the sequence of Ozempic, and then it has some other functional groups on there for solubility and stability and all these things. But again, if you know the binding site, you can synthesize a peptide that blocks an interaction, in this case blocks hunger and digestion and all of that. So huge markets, billion dollar pharma markets looking if you know the binding sites. If you don't know the binding site of the protein, it's a lot harder to target that protein, and there's really only two techniques that have risen to be very popular, and also both got Nobel Prizes in chemistry for them. So you can think about phage display, where you have all these phages with different random sequences of DNA that can code for peptides, and then you express the peptides on the surface of this phage, and you do like a pull-out technique to find strong binders. This is essentially what antibody evolution and discovery is. So a lot of the therapeutic antibodies that are on the market, like Humira, it's a top 10 selling antibody drug, it was developed through phage display. So this got the Nobel Prize in 2018. Now I'm getting my years. I think 2018. And then you can also think about computational protein design. So this would be like David Baker and the work at Google, where if you know the protein surface, you can essentially use machine learning and AI algorithms to develop binders for that protein surface. And this one got the Nobel Prize in 2021, I think. But both of these are really resource intensive. So phage display is resource intensive. Pharma spends a lot of money trying to develop antibodies that you don't, if you don't know the binding site. And protein computational design takes a lot of infrastructure, like a lot of computation along with protein expression and biological analysis and all of these things to find those strong binders. And while those lead to molecularly precise systems, they're very challenging to design binders for, and the scale up is really hard. Scaling up a peptide can be really expensive. Scaling up protein production can be really expensive, so you want to make sure that the target that you're using is going to be worth the scale up. So just some limitations that I find in this is that they rely on precise structures, either of small molecules or amino acids. This is great if you're going for therapeutics and you want to target a drug at picomolar, like you want a precise structure. But maybe if you don't want a precise structure, but you still want specificity, that could be a limitation because you can't make grams and grams and grams of oligonucleotides. Like that would be, I don't know if there's enough phosphorus to be able to make that much, right? And additionally, there's a lot of efforts to discover and optimize binding. Even if you're looking for something that's like an okay binder, there's a lot of work that goes into that. And one thing that I developed actually in my postdoc with Catherine, actually one of her mentors or mentees inspired this idea. But a lot of techniques are trying to get rid of a protein, but what if there's a protein there and it is like misfolded and like out of line? Can you just like put it back together? Can you just have a binder like that works as an allosteric activator that just puts it back together and then the enzyme works as normal? That's really hard to do. I'm a polymer chemist, so I'm going to talk about what people have done in polymer chemistry. There's a lot of other techniques to think about targeting and mimicking proteins, but we're going to talk about polymers. So you can think about, all of these are mimicking proteins for the most part. I'll get into interacting with proteins, but conventionally in polymer chemistry, most people are trying to mimic protein structures either through sequence design or property. So catalysis is very popular, where you have this sort of single chain nanoparticle where you have copper catalytic sites dispersed in the single chain nanoparticle. I'm just showing this azad alkyne cycloaddition where the rate of this reaction can increase due to the increased solubility of the starting materials in this sort of nanoreactor. This is a very popular area and has been for quite a while. You can also think about folding polymers to look more like proteins. This is one of my favorite examples from Abby Knight's group at UNC, where they put in these beta-alanine like structures where they could get this secondary structure folding within a polymer. So now you're getting a polymer that's starting to get this like protein-like secondary structure, which is really interesting to think about both from a design standpoint and like a way to understand more about protein structural dynamics. If we think more about like interfacing polymers and proteins, these are my two favorite examples. So Ting Zhu was a large inspiration for this work. This figure is essentially showing a protein surface where instead of looking specifically at like the chemical interactions on the surface, they zoomed out a little bit and looked at like big patches on the protein surface and said, okay, this big patch is cationic, this big patch is hydrophobic. Can we design a large polymer that can essentially like engulf this protein where we don't need sequence specificity because we're relying on like a length scale that's removed from like exact interactions on the surface, more like average interactions on the surface. So this is the science paper. Since then, they've shown a lot of work using these random heteropolymers. In this example, they took a protein that was engulfed in a polymer, moved it into toluene, and then they did catalysis in the toluene where the protein would usually be unstable, but the polymer was stabilizing the protein. So this is telling us that maybe you don't need sequence definition to interact with surfaces of proteins. Maybe we can think about different sorts of length scales and get averages of compositions. And then this example is from Nathan Janeski at Northwestern, where they're polymerizing peptides. So they're using ring opening metathesis polymerization to polymerize these peptide side chains. What they're doing is they're taking peptides that don't work that well, polymerize them to increase the avidity of the peptide, and now you actually have a really good drug. This is great and moving through clinical trials because essentially pharma has all these peptides that don't work that well. They can polymerize them and show that they actually make better drugs. A great idea. But what's really important from this is it shows you, even for clinical translation, the dispersity of the molecule may not matter that much if you have a good sell of why you'd want to do it. If you can take all the peptides and make a drug that you can't make any other way, or you can target some disease that you can't target any other way, maybe the molecular weight distribution from the polymerization isn't as bad as the benefit of treating that disease. So just to summarize some of the takeaways from here is that you can use statistical copolymers to mimic or stabilize proteins. A lot of chemistry is focused on mimicking. The Tingzhu work is thinking about stabilizing. Janeski showed that you can use these sort of dispersed copolymers and they still interact with the protein target. The polymerized peptide will still attach to its binding site and do the thing it's doing. So even though it's polymerized, it's still interacting with that protein surface in some level. But again, there's no way to modulate the function. So what we did is we took a step back and we're thinking about all of these examples. And a lot of this came from my time in the Merkin lab, just thinking about other ways to target proteins and thinking about interfacing them. And so what we did is we thought about a smaller length scale than Tingzhu usually works at. A lot of people think about protein mimicry. We were like, what if we try and mimic peptides instead? What are the length scales? What are the compositions? How much precision do we need to mimic a peptide that can maybe interact with a small portion of the protein surface? And what happens if we can do that? So in this first example that I'm going to talk about, we took green fluorescent protein. And what we did is we looked at the surface and said, okay, so there are these hydrophobic patches, highly polar patches, ionic charge patches, neutral patches. If we just look at the average chemistry of these patches, can we hypothesize acrylic monomers that would have complementary chemical interactions with these patches on the protein surface? So after a lot of trial and error, we came to these 4 monomers in this first paper where we're using this hydrophobic monomer for the hydrophobic patches, cationic charge monomer for the anionic charge on the surface, neutral polar for solubility and also some hydrogen bonding, and then just an acrylic monomer for hydrogen bonding. And the idea is if we can do a RAF polymerization, so essentially mix the monomers, bubble some argon through it, run the polymerization, and you can get grams of material. If we select the monomers well enough, we should get this ensemble of polymer compositions and sequences, and some percentage of that ensemble is going to have a high binding affinity for the protein as long as we're thinking about complementary chemistry to the surface. And so because we're chemists, we can think about how to test this hypothesis by changing composition, changing charge of the what we're calling random heteroligomers, changing the length of these oligomers from 6 mers to 48 mers. And we can take these random heteroligomers where we predict that some composition or some percentage of these are actually going to bind to the protein surface. And how do we actually study which ones are the strong binders versus the weak binders, or the winners versus the losers? And one of the reasons why we like green fluorescent protein is that you can monitor the fluorescence of the protein pretty readily. And we took, we know this is not the case anymore, but in the initial experiments here, we assumed that binding was going to be a direct readout of, or fluorescence was going to be a direct readout of binding. So if we're getting binding with fluorescence increase, that's an indication that the oligomer is binding to the protein and making the protein more fluorescent or work better than it does. Or you can think about denaturing where it's still binding, but we're denaturing the protein. So we get sort of a direct readout of what the oligomer is doing to the protein just by monitoring the fluorescence of GFP. I'm just going to talk about the winners. Darwin did a lot of polymerizations, different monomers to get here, but I'm just going to talk about these 4. And so the point of this slide, let me just ground you first in what this data means. So we're looking at the change in fluorescence over native GFP. So 0% would be native green fluorescent protein. If it is positive, that means that we're making green fluorescent protein more fluorescent than it naturally is. If it goes down, that means we're decreasing the fluorescence of the protein relative to the native protein. And then we have the different monomers with DHA and I, and then this number is the number of monomers. So this is an oligomer of 24. So with the cationic monomer, we actually get a little bit of increase in fluorescence. This is pretty widely known that cationic polymers can bind to like anionic proteins. That makes sense, right? But with the other monomers, they either don't do anything or denature it. But then as we start to go to binary, ternary, and quaternary monomer combinations, you see we're getting up to about 50% fluorescence increase. And it really depends on having these different monomers in this combination where we're getting the saturation. So like this combination of D and H maybe is giving us a pretty high fluorescence change. You may say these look the same. These actually have different dissociation constants. So we can actually change the fluorescence and make green fluorescent protein more fluorescent than we can express it to be fluorescent. And we can actually change the dissociation constant according to composition of the oligomer, which is really interesting. But the point of what we're really excited about is we really do need this combination of 3 or 4 different monomers to get these high increases in fluorescence of the GFP. So this is just looking at length, and I think this slide has one of the most important pieces of data on it, where if you look at just the monomer control at a pretty high concentration relative to GFP, just the monomers alone don't do anything to the fluorescence. So they're not changing the fluorescence of GFP if you just mix them. They have to be polymerized, which as a polymer chemist is amazing, because you're like, okay, well, you do need polymers to actually get this fluorescence increase. One other thing that I think is really interesting and important to point out on this slide, so we're looking at effective length, which I'll get to over here. But if we look at the GFP concentration that's at 100 nanomolar, if we add in 1-to-1 equivalents of our random heteroligomer to our GFP, we're still getting fluorescence increase. There are a lot of different sequences and a lot of different compositions in that we're adding. And even at a 1-to-1 polymer to GFP ratio, a lot of those polymers are interacting with the GFP surface and increasing fluorescence. So this is suggesting to us, for the binding that we're targeting, we don't need sequence precision to still see a functional output. We can look on average compositions and still see a functional output, which is really exciting to us. The data on the right is just showing that it's an effective length, which makes sense. So the fluorescence saturates around a DP of 24, so 24 mers. This is looking at apparent dissociation constants. Again, it sort of plateaus around 24, and it doesn't get much higher than that. So all of this is saying is that we can actually have a pretty short polymer or oligomer where we can get fluorescence increases and pretty good dissociation constants. This is about 100 nanomolar to ground us, and what that means, a bad antibody binds at around like 1 to 10 nanomolar. Good antibodies are in the picomolar. We're like in the range of a bad antibody, which is fine. You know, this is a single polymerization. Like you just mix the monomers, do the polymerization, get your polymer. We're getting like 100 nanomolar dissociation constants, which is really interesting to us and incredibly scalable as well. I like pictures. So this is just a picture making sure that it's not just due to molecular crowding for this fluorescence increase. So this is GFP under UV light. So GFP just looks like that. We added in a polyethylene glycol control of the same molecular weight and same concentration just to make sure it's not crowding. And then as you add in your DPs, you see a very visual difference in fluorescence. Depending on the projector, you can see that the DP24 is maybe a bit brighter than the DP12. So even like an effect of the length, you're seeing the brightness of the GFP, and it's very visual that the GFP fluorescence is changing even like on the benchtop. We also ran some native electrophoresis. So what we're doing here is, because it's GFP and we aren't denaturing it, we're imaging the fluorescence of the GFP in the gel. So we can excite GFP and measure fluorescence on our gel scanner. So if we just look at GFP in this native gel, as we increase concentration of our random heteroligomer, so all of these lanes have the same amount of GFP in them, we're just changing the amount of rho. You see that the band gets darker and darker, meaning that we're increasing the fluorescence of that protein in this native electrophoresis experiment. This is cool because it means that these complexes are stable under electrophoretic conditions that also suggest that they are actually interacting with the protein surface and they're relatively stable. In this case, we normalized to GFP fluorescence now, so we essentially calculated how much GFP to load with the random heteroligomers, where all the bands would be about the same intensity. And if we normalize to GFP fluorescence, under the presence of the rhos, we actually see a slight shift in mobility, which also suggests to us that the rhos are binding to the protein surface and sort of changing the surface charge, surface properties, so they move through the gel differently. So all of this suggests to us that the oligomers are interacting with the protein surface. We did a ton of other characterization to confirm that, but I skip over some of the characterization just for time. In the paper, there's a ton, so if you want more, look at the paper. So where we've gotten so far is that you can take this target protein with your random heteroligomers, and you can change composition, charge, and length, and increase fluorescence. But the big thing is, like, we're just doing a single polymerization with so many different sequences, and we don't actually know what the winners and the losers are in this composition stoichiometry that we're adding. So what we did next is we took our polymerization and we shot it through an HPLC with a C4 column or C18 column. And so by fractionating by hydrophobicity, we can actually collect different fractions of that single polymerization and understand how the composition from that polymerization is actually affecting the binding and whether there are like stronger binders in solution compared to weaker binders in the solution. Again, this is a single polymerization from around bottom, shoot it through an HPLC, and we fractionate it that way. So the left one is just showing the HPLC fractions. So we took fraction 1 through 8, and we characterized them by gel permeation chromatography. All of the fractions overlap with molecular weight distribution, so all the fractions are about the same molecular weights, same dispersities, all of that. If we characterize composition by NMR, we see that the fractions increase with hydrophobicity as the fraction goes on. This makes sense. We're just doing affinity chromatography with a hydrophobic column, so the hydrophobic things will elute later than the less hydrophilic thing, or the less hydrophobic things. So fraction 8 has about the same amount of cationic charge and hydrophobicity. And then as the fractions go on, the hydrophobicity increases and the cationic nature decreases, whereas the other two monomers stay about the same. And now if we do assays with these fractions from the polymerization, we actually see differences in functionality even with those fractions. So this is saying like, even though as an ensemble we're getting increases, there are like preferential binders within that single polymerization, and we can actually isolate them and characterize them. So this is really cool because this is equivalence to GFP. So essentially if our GFP is at 100 nanomolar, we can add in 250 nanomolar of our random heteroligomers, and we get essentially fluorescent saturation for a lot of these signals. So that's a pretty low equivalence of binder to protein where they're not sequence defined, and we have different compositions. Even though we enrich them, we're still getting pretty strong binding just by the composition of monomers that we added. And then as we go on, we see the hydrophobic ones don't bind as much at these low concentrations of oligomer, but if we add 20x in, it's a little higher. We can also show that from the crude polymerization, we can split the apparent dissociation constants to be stronger binders and weaker binders. You'll notice these values are a little different. I just want to talk about that for a minute. So we've done all of this data from start to finish with 3 different protein batches and 3 different polymer batches, and it always depends on the protein batch. So the protein batch is the thing that dictates the apparent dissociation constant, the fluorescence increase, all of that. The polymers behave the same every single time that we make them, which is really interesting to us because we think they're actually sort of affecting the folding and all of these things. But we're looking into that and more to come there. But this is really interesting to us because it shows that we can pick some winners and losers. So the next thing that we did is like, okay, we can isolate this composition, but like what's actually happening in the binding? Like how are these oligomers actually interacting with the protein surface? That's really hard to characterize. So what we did is we took a composition from the strong binder, a composition from the weak binder, randomly generated sequences and ran computational analysis on them. So this is from my colleague Sanket and his postdoc Seth. And so this is a heat map of the protein surface. So green fluorescent protein is a beta barrel protein. You can look at this as like unwrapping the protein, and then you have a heat map of binding of where the oligomer is binding on that surface. So it's like a 2-dimensional heat map of binding. So for the strong binders, we can see local binding minima. We can see about 6 of them on the green fluorescent protein surface. The barrel is essentially right here. So you can see that there are 6 points on the protein surface that these oligomers preferentially bind to during these sorts of simulations that we're running. If we compare that to a low binder, you see that maybe there's a minima here, there's a minima down here. This is a coil region, this isn't in the barrel region. But just by looking at these, you can see that the strong binders, depending on composition, show a lot more binding sites for the GFP surface than the low binders. We ran maybe 20 different sequences and compositions with this sort of computational analysis. And then what we did is we took these poses, because we're running, these are coarse grain, or these are essentially like putting a protein in a box, and then the oligomer like moves around the box, and you can tell it not to bind somewhere. But then what we did is we took the poses from these, and we ran high-level polarizable molecular dynamics with my colleague Valerie Welburn. So in this case, this is the high binder, so you can see the protein in blue. This is the oligomer, it's interacting on the barrel. This is the low binder pose from the last case where the protein is in orange, and the oligomer is down here at the bottom of the coil that I was talking about. And so these are 100 nanosecond simulations. They take about a month and a half to run. And what you see is that the high binder stays around the barrel and is mostly interacting in the same site, whereas the low binder is going to fly off and not interact with the protein again. So even in these sorts of high-level computational analyses, we see that this oligomer is interacting with the protein surface around the barrel. And from the metadynamics, we know there are multiple sites on the protein surface that these oligomers are interacting. What's interesting is that that number of sites actually correlated pretty well with what we found experimentally on the number of polymers per protein. But this is really exciting to us because we are seeing like compositional differences in these sorts of high-level computations. We've ran a couple different sequences. I think we've done a total of 10 simulations here. But again, these are very computationally expensive to run. So we haven't run as many as the metadynamics. So we're really excited about this. And this is showing us that we can use these random heteroligomers. I just want to highlight where we're going next. I'm not going to talk about all the data. But essentially, what we're trying to do is think about a high-throughput machine learning way to do this, where we're not like Darwin isn't in lab like crunching out all of these polymers. We can actually predict better. So we worked with BioPacific MIP, and Darwin went to BioPacific MIP and used their robot to synthesize 1,000 different polymers in 4 days with 11 different monomers, and then he did 4,000 protein binding assays. This is just a layout of what a 96-well plate looks like, where we came up with a specific design where you can have 4 different monomers, and then you change composition and length within this 96-well plate. Then you see we get these fluorescent readouts from the assays, and then we can use all that data to learn like structure function properties. This is just some of what happens. So these are all those experiments. These are 1,000 different polymers, and you can see most of the polymers that we synthesize don't do anything. We have some that destabilize, we have some that stabilize some, and then we have some that are increasing fluorescence of GFP quite a bit. And all of these are novel compositions that we haven't done before. So we're working on finishing up this work. It's really interesting to think about. And then the last thing I'll talk about briefly, but not give any data for, is we took all of this. And now what we're doing is working with Sonkit to put it into a like agentic AI system where we have essentially designed and collected all the data. We have a ton of data that we have tested. Sorry, start down here. So we have a ton of data that we've tested and gotten the assays. And now what we're doing is we're developing this agentic AI system that is trained on all of that data. And then we challenged it with essentially every acrylate and methacrylate that you can buy. It spit out new compositions for us that we haven't tested at all, so we're testing and validating those systems. And then in the simulation, or in the AI design, it's also coinciding with essentially like a docking experiment. So we can predict compositions to get fluorescence, and then we can also combine it with docking to get like binding distinct from fluorescence increase. So you can think about like strong binders that decrease fluorescence or weak binders that increase fluorescence, and you essentially have like this tunability between fluorescence and binding. And we're also trying to expand this. We're trying to think about just the chemistry of the protein surface, not the protein, so that can we like expand this to other proteins by looking at specific patches in the composition of those patches instead of like whole protein. With the goal of being able to like have the system where you can just say, I want to target this protein, and then it spits out a composition with this sort of AI docking backend that happens. So we're really excited about that. It's getting there. It's almost done. It's almost done, but I didn't want to talk about it today. Maybe next time. But essentially in this section, the whole idea is that we can take this protein and look at sort of we're not looking at exact sequence. We're like zooming out one level and looking at average composition on patches, designing synthetic polymers that can target those patches based off complementary chemical interactions. And again, we're not trying to make antibody replacements. We're trying to think about areas where you want scalability and relatively good specificity. So you could think about chromatography techniques, maybe like rapid tests. There's a lot of antibodies that work well, but not well enough for therapeutic translation. So can we like pre-select out some of the competing proteins? So that's the thing that we're trying to think about, like how do we complement this with other techniques that may be more precise. So I'm just going to finish there and thank my group. This has been supported in a variety of different funding mechanisms, and all the students that did the work were on the slides. And I just want to highlight, I think both of the MIPs are now open to international collaborators. Both of them are great. GlycoMIP is at Virginia Tech, and then BioPacific MIP is at UCSB and UCLA, and they have a ton of infrastructure for like automation and assays and glycomaterials. And they've supported that second part of the project a lot. So with that, I'd love to take any questions.
Very nice talk. When I see this like 24 like these polymers with 24 monomers inside and based on 40 polymers. Like from what I see, you need to do a large synthesis and then collect fractions to get the sequence that you are most interested into. So like, what is your strategy to work towards more precise synthesis to have less byproducts or like sequences that are not interested in?
Yeah, so essentially the question is how do we make them more precise?
Yeah.
So the first half of that talk is we're not doing any purification, we're just taking the polymerization that we get. We can think about synthetic strategies to make them more precise, but I think the point is all of those are more cost intensive. And are there ways that we can think about composition and very low barrier ways of purification? Because we don't want to make synthetic peptides. There's a ton of people that are doing that, it's very expensive, it's not scalable. We're trying to think about like how much precision and selectivity can we get in something that's really scalable. So in like the use case, we're actually trying to think about how to use the chemistry to make it more selective, but we don't want to go through a bunch of purification because that defeats the purpose of the idea. Because like if you want a really precise molecule, you can make a peptide or a peptidomimetic, but if you want something that's scalable and relatively precise, we think this is a good technique. But we're making them more precise to understand the interactions. So like as an academic lab, we're interested in doing it and using some polymer chemistry techniques and polymerization techniques to understand it, but that's just to inform the design of the less precise ones. But ideally, like you could just make kilograms of this material, do minimal purification. Like, you're not gonna get selectivity for every single protein because we're not making molecularly defined, but we're not trying to do that. We're trying to just select for the proteins. Like you can think about it like a pre-screening. Yeah.
But here is because like between your low binding and your high binding one, at the end of the day, whatever the precise sequence is, you have two monomers that are truly different in the polymer that you have, right?
They all have the same compositions, or they all have the same 4 monomers. They're just at different ratios.
So yeah, but like here in your ratio, it's like at the end of the day, you had one that was 4 and 4 and one that was 2 and 6 or something like that. So
Yeah.
this level of precision, and so
Those are averages.
yes.
Yeah, those are all averages.
Okay.
So we're not getting, like even the high binder, we did the calculations and there's 10 to the 18 different sequences in the high binder. So there's a ton of variety. Those are just averages. But we're just saying like with more hydrophobic ones, those hydrophobic interactions don't like prohibit ensembles of oligomers from binding. We're just, if we have this broad distribution, we're isolating ensembles, but it's still a ton of variety. Yeah. But we're not trying to make like a sequence-defined acrylic polymer because you can't do that. But like what can we do with the techniques that we have now that can actually have an impact on like health materials and protein binding? And like where are the use cases where that could be useful? Yeah. Yeah.
So in the second half of your talk, you're basically talking about how you're making these random copolymers almost. And they kind of have that random sequence because they all form a secondary radical, right?
Yeah.
So my question then is, going back to the first where it was on, let's say you were to do a rapid polymerization in that same way, but you have like you have an acrylic polymer and an acrylic polymer. So methyl acrylate and methyl acrylate. What do you think the sequence of that resulting polymer would it be? Would it be closer to like a copolymer, or would it be kind of like?
Let's see. Do I know the reactivity ratios? I think the reactivity ratios are like somewhere in the ballpark of like 4 to 0.3 or something. So like a methacrylate is like 3 to 6 times more likely to react with itself than acrylate, and an acrylate is 4 or 5 times more likely to react with a methacrylate. So based off the reactivity ratios that you measure, you can predict like average sequences and compositions and get like blocky or gradient-like statistical copolymers. My grad student, Michaela, who's in the front, that's what she's doing right now. So we're starting with, so this may not have been obvious, but one side of my group is trying to essentially make more defined sequence-defined polymers. And then the other side of the group is saying, does it actually matter if we make sequence-defined polymers? So we're trying to meet in the middle. And so both of those are always in conversation. So Michaela's more she's interested in the proteins, but wants to think about sequence precision. But we're always asking, like, does it matter at the same time?
That's really cool.
Yeah.
Thank you.
Yeah.
With regards to your protein bonding polymer formulations, have you ever ran into issues of denaturation or aggregation? How often does that happen? And in particular, with a green fluorescent protein example, are you able to differentiate between whether you just get fluorescence quenching or if you get true denaturation or aggregation?
Great question. Okay, so in the data that we've talked about, we don't see aggregation. So one of the things that we're wrapping up that I think is super cool and I'm very excited about is we can actually, like, when you express the green fluorescent protein that we express, it gets expressed in inclusion bodies, and then you have to centrifuge it and then like denature it and renature it. We can just add our oligomer to the pellet, and the oligomers will pull the green fluorescent protein out and fold them. So we think that they're helping solubilize the GFP with the composition. What the computational data is showing and what we've observed in the lab, actually, I got this question when I was giving the seminars, when I was like, oh, it's denaturing, and they're like, how do you know that it's not just binding really strongly and denaturing? And we're like, oh yeah, duh. So that's why we actually did a lot of this machine learning, so we can actually target denaturing. So essentially what we think is happening is that the oligomers are binding to the surface. The ones that we have, they're binding to the surface with enough binding affinity that they're sort of restricting the thermal oscillations of the proteins. Then the chromophore fluorescence goes up because they can't thermally relax. So we're increasing the quantum yield of the fluorophore. But if that binding is too strong and the oligomers can get into the barrel and break it up, then you give a lot of flexibility to the chromophore because the protein is denatured. So that's what we're starting to look at, is like, and the computation that I didn't talk about is there's actually a sweet spot in the machine learning of like binding and fluorescence increase, and then if it's too strong or too weak, other things happen. So we're starting to look at it and think about it as like tunable. Yeah, but experimentally, sort of, but we just kind of got, I don't like saying lucky because I don't think, we selected the right monomers through 20 other monomers that didn't work that were giving us the fluorescence increase, but now that we're thinking more about what's happening with the protein, you can sort of toggle it.
That's good, thanks.
Yeah.
I want to see if you can make polymer chaperones.
Yeah, agreed. Yeah, we don't have like a ton of, we have good specificity, but it's not great. So, you know, we're limited in sequence. So like how good can we get it and where are the uses where it's good enough is what we're going for. Yeah.
Well, thank you so much, and please join me in thanking Professor Figg for the seminar.